Hello everyone, my name is Zhen Han. I am the Key Accounts Deputy Director of International Sales Department at Fapon Biotech. In today's webinar presentation, I am going to discuss with you some of the technologies that we have used at Fapon Biotech to develop high-performance raw materials for IVD applications. Before starting the presentation, I would like to thank Alice Liu, who is the head of our antibody discovery department. Alice had helped a lot in preparing for the slides that we're going to discuss about in the next 30 minutes. There will be 4 sections in today's presentation. Firstly, I'll do a brief introduction of Fapon Biotech. In the second part, I'll introduce Fapon's raw material development technologies at a more detailed level. In the third part, I'll present some case studies to demonstrate how do we apply these technologies. Lastly, in the fourth part, if you're attending ADL in Chicago this year, I would like to invite you to visit our booth at 3816. Without further ado, I'll get started with the company introduction. Fapon Biotech was first established in 2001 as a raw material supplier company, focusing on developing antibodies, antigens, and enzymes for IVD applications. Over the past 2 decades, we have expanded our business portfolio and providing not only raw materials, but also reagents and open system instruments. Specifically, we have made great sales on our chemiluminescence immunoassay analyzers, both in China and globally, and received many positive feedbacks from our users. Our sales growth of the Clear analyzer had been ranked #1 in China in 3 consecutive years in the past. As of today, we are 24 years in business and provided the industry with over 1,400 products across 3 major different platforms, including chemiluminescence immunoassays, lateral flow rapid test, immunoturbidimetry assays, and we're also providing products on the molecular diagnostic field as well. We have filed over 1,300 patent applications around our products, and we have worked with over 2,500 customers worldwide from more than 70 countries and regions. To work with the community around the world for pandemic prevention, Fapon recently joined the Pandemic Fund, and we look forward to working with the partners to build a healthier environment. Last year, we were also proud to be named as one of the top 30 selections of Go International Brands by Forbes China, which highlighted our achievements in international market expansion, innovation, as well as brand influence. In addition to that, our commitment of building a sustainable and a responsible business is also recognized by EcoVadis, which ranked us top 35% in their recent sustainability rating last year, 2024, in November. Not only do we have broad product portfolio, but also geographically, we have built 5 R&D centers around the world. Our headquarters is based in Shenzhen, China, where we're developing and manufacturing open system instruments for chemiluminescence immunoassays as well as molecular diagnostics. In Dongguan, China, we have built a R&D center as well as manufacturing facilities to develop and manufacture our IVD raw materials and reagents. We have also built R&D center in Shanghai, China, where we are focusing on biopharmaceutical and AI development. In the United States, we have R&D centers in Boston and San Francisco, focusing on development of molecular diagnostic enzymes, as well as NGS instrumentations, respectively. Being able to locate at biotech centers around the world really help us to stay innovative and progressive, and help us to build better and better products to help our customers. Within each site, we've also built high-standard R&D and manufacturing facilities. We have over 100,000 square meters of facilities globally that is currently being used. Below are some pictures of our lab spaces dedicated to different applications, such as antibody discovery, stable cell line development, high cell density fermentation, protein purification, QC, as well as you can see some chemiluminescence immunoassay analyzers at the bottom right corner. The next slide is about our dedication in quality management. Fapon values quality since day one we established. We're actually the first IVD raw material supplier company in China, obtained ISO 9001 certificate. Subsequently, we have obtained ISO 13485 certificate, which highlighted our commitment in maintaining high product quality. and the ISO 45001 and 14001, which showed our dedication in building a safe and environmentally sustainable workplace. We're also recognized as one of the AEO Advanced Certified Enterprise, which in many cases help us to perform international businesses more smoothly and efficiently. In our daily operations, we also apply stringent quality management system, all the way from incoming QC to final product QC and troubleshooting. We make sure each step of our product development and sales activities are well managed and traceable. We also established a robust patent filing system. So far, we have filed over 1,300 patents, covered not only China, but also other countries and regions we run our businesses. 500 of the patent applications have been granted, and 80% of our patent applications are for inventions, which really highlighted our dedication in promoting innovation. But more importantly, the patents enable us to better leverage our proprietary technology to help our customers in a very sustainable way. All right, now let's move to the second part, which I'll be presenting Fapon's raw material development technologies. This slide summarizes the core technologies that we have implemented in our raw material development platform. It included protein production, purification, QC, antibody discovery, engineering, as well as asset development. On the protein production side, we routinely implement different expression systems, including mammalian cell expression, yeast expression, insect cells, and bacterial cells, such as E. coli. We have also developed a high cell density fermentation process for large-scale manufacturing, as well as a high-throughput antibody purification for antibody screening at early phase. And specifically for inclusion body purification, we have developed several refolding technologies. During both R&D and downstream manufacturing processes, we apply optimized purification methods and standard QC processes to monitor our product quality. For antibody discovery, we have developed 3 different technologies that we mainly use, including the single B cell platform, the hybridoma platform, and phage display platform. We can also perform antibody engineering using structure-based analysis, affinity maturation, as well as humanization. Lastly, the asset development, which is also a critical component for the raw material development. We routinely test the compatibility of our raw materials on different asset platforms, including chemiluminescence immunoassays, lateral flow rapid test, and immunoturbidimetry assays. From our experiences, we have developed a good number of optimized SOPs for key asset development steps. All right, after the overview of our raw material development platform, let's look at the technologies closely to see some of the advantages we can offer from each sectors. The first workflow or technology I like to go through is the protein production and characterization workflow, which covers both antigen and antibody productions. Taking antigen as examples, as many of us know very well, either as immunogens in antibody discovery campaign or calibrators or quality control in the assay, high-quality antigen material are very important to have. However, antigen expression can sometimes be very challenging because of their low solubility, inclusion body formation, or complex post-transgenic modifications. We have run into such situations quite a lot in the past, where we needed to do some engineering so we can redesign the protein to make them more soluble and stable during the production processes. That is when we accumulate a lot of experiences. We usually implement the engineering when designing the gene sequences and the molecular cloning methods. We mainly use site-specific immunogenesis, protein truncation or hybridization if we need to alter the antigen sequences for expression. We also have a library of over 200 fusion proteins we can co-express with the target protein to improve their stability or solubility during the production process. And sometimes we use the fusion protein to improve the immunogenicity of the target protein in an immunization campaign. We have also developed over 100 expression vectors that are optimized for different expression systems for high gene expression level. The last step of our construct design step is the codon optimization. By doing so, we make sure that we use the right genes for the right expression systems. For either antigens or antibodies, after getting the vectors, we move to the expression and purification stage. Using different expression system, as I've mentioned before, we routinely perform the transient transfection and stable cell line fermentation at different scales at different stages of that project. We can express protein at high throughput in small scales for affinity screening, and for the final selected raw material, we can also scale up at a manufacturing scale. While working on the scaling up and affinity and functional test, the downstream process development focusing on establishing a purification workflow is also up and running in parallel. We have different purification methods such as affinity chromatography, refolding technologies, and we also have some polishing steps such as anion exchange, hydrophobic interaction, or size exclusion chromatographies, and various filtration methods for the final product. We can also conduct formulation development with the final product and make sure that we use the optimal buffer for the long-term stability and function of the protein. During the development process, we routinely use our analytical toolbox to analyze the physical chemical properties of the protein, such as their size, their charge variance, as well as aggregation. We also conduct binding assays such as plate assays and SPR to look at the proteins from a functional perspective. To summarize, we routinely apply this integrated process development workflow for transition of raw material from early discovery stage to a robust and manufactured product that is ready for preclinical and clinical studies. Next, I would like to introduce our antibody discovery platform, in which we have implemented 3 major technologies, including the hybridoma technology, the phage display technology, as well as the single B-cell sorting technologies. As is shown in the table, each method have their own advantages. Depending on the goal and the timeline of a project, we can choose either one or several of these methods to ensure a success of a project. We're also doing some internal research to develop an artificial intelligence-based method to identify antibody binders, which can significantly shorten the turnaround time for antibody discovery workflow. In today's presentation, I'd like to mainly focus on the phage display and single B-cell platform. Compared to the hybridoma technology, which is more traditional, these 2 methods offer higher efficiency and shorter timeline in developing recombinant monoclonal antibodies. First, the single B-cell workflow. I would like to show you the end-to-end raw material development process using single B-cell method as an example. The process begins with the immunization. We usually use rabbit because it's strong immune response. We have established 2 major immunization methods, one traditional method and one express method. The traditional immunization method usually need 8 to 10 weeks, versus the express immunization method requires only 4 weeks for the animal to establish strong immune response and make the target antibodies. Following the immunization, we monitor the serum antibody titer through an ELISA assay to confirm a strong and specific immune response before we proceed. Once we have observed a high enough titer, we move forward with B-cell isolation and recovery. In this step, we use an in-house developed proprietary medium to increase B-cell recovery efficiency. After the B-cells are sorted or isolated with the labeled antigen, we will perform a round of titer testing, which is the screening stage. The supernatant of B-cell isolates will be tested for antigen binding using ELISA assay. After the sequence is down, we'll move forward and sequence the positive B-cells. We'll get the antibody sequences for downstream recombinant expression. We usually analyze hundreds of thousands of sequences in the cluster format, so we can choose the antibody sequence that are relatively more distinctive from each other. This way, we have a better chance of getting antibodies that can cover different epitopes of an antigen protein. This is basically the last step of the antibody discovery phase. Once we obtain the antibody gene sequences from discovery, we begin the process development step. The process development involves optimizing expression constructs, the cell line, the upstream and downstream workflows, followed by a scale-up for manufacturing, which I've gone through this in the previous slide. Once we have narrowed down the raw material to smaller selections, the final evaluation is performed using the final intended assay format. In this case, it could be a chemiluminescence immunoassay, lateral flow colloidal gold assay, or immunoturbidimetry assay. We're running this test to confirm the compatibility of our raw material on these assay platforms. We'll be testing the sensitivity, specificity, and overall performance of the assays. And this step is really to ensure that our antibody and antigen raw material functions optimally on these platforms before we deliver them to our customers. Next, I would like to introduce another antibody discovery technology that we have implemented, which is the phage display. This is a great approach we often use complementarily to the single B platform. There are different host species we can use to build immune libraries for panning campaign, including mice, sheep, rabbit. In the case of if we wanted to build or develop single chain antibodies, we can also use alpaca. During the panning stage, we can use diverse antigen types and formats. The antigens could be proteins, nucleic acid, small molecule, or peptide. We can also immobilize the antigen on different surfaces, such as the beads or plate. We can also overexpress the target on cell surface if, for example, if it's a transmembrane protein. Basically, we'll select the optimal format for the best condition for the protein to demonstrate epitope and fold properly in that environment. After each round of panning, we sequence the enriched phage libraries to monitor the clone enrichment to maintain the library diversity. By tracking the sequences from round to round, we can ensure that we're capturing the high affinity antibody gradually without losing the diversity of the phage library too early in the process. The top candidates are then sequenced and selected after the final round of phage elution and followed by the steps the same as is for the single B cell workflow. In the next slide, I would like to discuss our bioprocessing workflow, mainly focusing on the upstream side. On the left side of this chart is our stable cell line development workflow, which usually takes about 2 to 3 months for us to develop a stable cell line, all the way from construct design optimization to the cell line stability test. First, for expression construct design, we usually select the appropriate signal peptide, the vector backbones, and the promoters to ensure high transfection efficiency as well as high transcription and translation efficiencies. The stable cell line development started with the whole cell transfection, which we have different methods we can use for the optimal result, including electroporation, lentiviral transduction, as well as transposase-based methods. After the transfection, we'll apply selection pressure, and we will usually screen hundreds of thousands of clones to identify a high-producing cell line. After that, we will also test the stability of the cell line, usually over a passage of 30 generations, to make sure the gene expresses stably across the time. In parallel, we also optimize the culture condition, including the media composition, the feeding strategy, the bioreactor parameters such as the temperature, pH, dissolved oxygen, to maximize the yield while maintaining the product quality during the process development. We have different fermentation sizes, ranging from 1 liter all the way to 1,000 liter for process development and manufacturing respectively. We have a total of over 2,000 liter manufacturing capacities, which in most cases enable us to support several customer projects simultaneously. In the next slide, I would like to show you our comprehensive assay platforms. We are able to routinely evaluate the performance of our raw materials on different assay platforms, as I mentioned before, including chemiluminescence immunoassays, lateral flow rapid tests, as well as immunoturbidimetry assays. Our team brings deep experience in key optimization steps, such as antibody pairing, conjugation chemistry, buffer system design, as well as matrix effect minimization across the 3 major assay platforms. For example, in the chemiluminescence immunoassay, we can help you optimize the signal-to-noise ratio of the assay by fine-tuning the AE labeling and magnetic bead coupling. For lateral flow, we can guide you through the selection of the gold particle size, the membrane type, as well as the flow rate. And for immunoturbidimetry, we can help you customize the latex antibody coupling. We can offer all the way from raw material to semi-finished products for these 3 platforms, which can significantly shorten the development timeline for our customer and help them to accelerate the product launch as well as commercialization. In addition, I would like to quickly go through some of the physical, chemical, and functional analysis tools that we have constantly using internally for our product development, some of which have been mentioned before in the previous slides. For example, with the protein sample and analytical HPLC, we were able to evaluate protein purity, protein size, and charge variance and aggregation, etc. The plate-based binding assay, of course, used throughout the entire process when we need to develop the antibody antigens. And we also have the capacity to look at the kinetics of antibody antigen binding using the Biacor system on SPR assay format. Now that we have walked through our core technologies and development capabilities, I would like to show you some of the key studies to demonstrate how do we apply these technologies in real-world examples. In the first case study, I would like to talk about the development of an antigen rapid test targeting on influenza, specifically flu A. As we know, flu is a significant global health burden, causing seasonal epidemics and results in millions of severe cases and hundreds of thousands of deaths each year. Because of that, the rapid and accurate diagnosis of flu is very essential. One of the key challenges in developing reliable flu diagnostics is the genetic variability of the influenza virus. Frequent mutations, particularly in the surface proteins such as HA and NA, lead to emergence of new strains each year and each season, making it really difficult to capture all of them with high sensitivity products. To overcome this issue, our assay targets the nucleoprotein, which is a highly conserved protein expressed internally by both influenza A and B viruses across different strains, which makes it a very ideal marker for broader detection of the flu viruses. To further increase or enhance the coverage of our assay, we did some sequence alignment and a bioinformatic analysis, and we engineered the N protein sequences to capture all the conserved epitopes across multiple circulating strains at a time. After the sequence is designed, we quickly moved to antigen production, following the protein production and purification workflow. Also, the process is shown on the right side as well. After going through and optimize some key steps from transformation all the way to downstream development, we were able to successfully manufacture this protein with a pretty good yield and function. In the next step, we move forward to the antibody discovery. In this particular case, we used the single B-cell sorting method. We used the manufactured N protein as an immunization, and following immunization, we've seen robust immune response from the rabbit via titer test. For single B-cell isolation, in this case, we first removed the cell debris and the dead cells, followed by a B-cell labeling with bioattenuated N protein. The labeled B-cells are then subjected to isolation and culture using an internal designed medium. The antibody titer was then tested using ELISA assay. For this particular project, we were able to screen through around 6,000 clones, and we were able to find more than 2,000 clones with the antigen-specific binding affinity, which is about 40% positivity. And then we moved on to amplify the sequence of the antibody variable regions from the positive clones and managed to sequence 95% of them. So based on the affinity ranking and sequencing cluster analysis, we picked the top 96 antibody for the next step, which is antibody pairing and screening. Because in this particular case, we're developing a sandwich assay for antigen test. So we need a capture and a detection antibody for co-recognition of the N protein. We have built a workstation for this purpose. As the system shown on the left side of the slides, it's a workstation that helps us to screen 96 antibodies in the matrix format in about 3 to 4 days, and it kept us from doing all the tedious manual processes, which help us to reduce the timeline and also minimize the potential human errors. As you can see in the heat map shown in the middle, in which the capture and detection antibodies are represented horizontally and vertically, respectively, we were able to find some really nice capture and detection antibodies, as well as strong antibody pairs, which can be represented by the darkest color spots. Those pairs of antibodies were then moved to the next step and validated on the final assay format, which in this case is a colloidal gold lateral flow assay. Eventually, we were able to develop a really robust fluid combo assay using antibodies we have developed from this project and also some flu B antibodies we have developed from another project. Our assay showed great analytical sensitivity, as you can see on the top left corner. Our assay can detect all the strains from the NIBSC and Zepto matrix panels. We also compared with our assay with the PCR method as well as some other rapid test assays on the market, and we have observed great concordance and clinical sensitivity of our assays. As shown on the top right corner, we were able to get very high PPA and MP values in the study. As shown on the left bottom corner, we have also conducted cross-reactivity check, and we have observed no cross-reactivity with our assay when testing against some relative pathogens as well as drugs. As a result, a good number of IVD companies around the world from different countries used our raw material to develop their own flu or flu COVID test, and they have achieved great commercial success. Now I would like to discuss another case study about how we developed a small molecule salvage assay in this particular case for vitamin D detection. As we know, vitamin D deficiency has been linked to a wide range of health issues, which makes it a critical and high demand target in clinical diagnostics. However, due to the small size and strong hydrophobicity and low blood level of 25-hydroxyl vitamin D, developing a robust assay becomes quite challenging. Traditionally, most vitamin D assays have relied on competitive assay formats, which can suffer from low sensitivity, narrow dynamic range, and most importantly, it's very easy to be interfered with some interferences. To tackle this problem, we developed a salvage assay for vitamin D detection. The key for the success of this project is to develop the right antibodies. And fortunately, we were able to do so following the similar workflow as I've shown before. And on the right side is our assay protocol, which has been validated on the chemiluminescence immunoassay analyzers that we have developed internally. But the assay can also be widely implemented on other clear systems as well. Now taking a quick look at the assay performance, our assay showed great sensitivity with the LOQ at 1.5 nanogram per ml. The assay range is between 1.5 to 150 nanogram per ml, which is well covered the entire pathophysiological range of vitamin D detection. We have also seen high correlation of our assay when compared to the control method, which is LC-MS-MS, as well as some other products on the market. In addition, our assay employs a single component lysis buffer, which is simple to manufacture and also is very effective in releasing the total vitamin D, including vitamin D and D3 for detection. The interference and stability data is not shown here, but the results looks very good. If you're interested in testing this product, please feel free to reach out to us. I hope I was able to clearly present the application cases of our raw material development technology. And with that, I wanted to wrap up the webinar by thank you for your time and attention. If you have any questions regarding the webinar, please feel free to reach out to us. Our marketing team is ready to help you, and also you can reach out to me directly. Last but not least, as I mentioned before, we're attending ADLM in Chicago this year. If you're also attending, we want to welcome you to visit us at booth 3816. We look forward to seeing you there. Thank you very much.